Right, having Dr. Chris Wilson here to give us this lecture on rotifers is a result of a remarkable coincidence, really. When Martin and I were talking over a sort of draft list of possible presentations, we both agreed that it would be wonderful if we could have something on freshwater invertebrates, and especially rotifers, because this event has been partly funded by a small legacy that we were left by Eric Hollowday. And Martin suddenly then, I think, emailed me or said that he'd recently witnessed a very interesting lecture in Oxford, I think, by Chris. And why didn't we try and get him? Well, I kind of assumed it would be very, very difficult to get somebody as eminent to come and talk in Great Lissenden. But Martin used his charms, I think. And here we have Chris to talk to us. And then we suddenly thought, well, let's call it the Eric Hollowday Lecture. Let's make a thing of this. And on the back of that, we've also had Charles Hussey over there, who's put together a fantastic display. Just make sure you go and look at that. Look at the amazing detail in Eric's drawings and his little timeline of his kind of involvement. Absolutely fantastic. So really pleased to have Chris here to sort of celebrate really a very important member of our group for many years, but also tell us about, I have to say, a group of invertebrates that I know next to nothing about. So over to you, Chris.
Thank you very much. Good afternoon, everyone. I should first say that I'm really honoured to be here, to be invited to join you and to speak for today's inaugural Eric Hollowday Lecture. It doesn't take much to persuade me to twist my arm to come and talk to people about rotifers. So they're a group that I've studied for 20 years or so. I'm a lecturer in biology at St Hilda's College at the University of Oxford, and my research is in the genetics and evolution and a little bit of the natural history of bdelloid rotifers in particular. I'm going to share some stories about these fascinating little animals. As I'm doing so, I'm sort of acutely aware that Eric Hollowday probably knew far more than I do about the taxonomy and natural history of rotifers. So I hope, and there's at least one other eminent expert on the taxonomy of rotifers in the audience, so I hope they'll forgive me for any small taxonomy, natural history and identification mistakes I might make along the way. So I don't think I ever met Eric. We may have overlapped at a rotifer conference once in Berlin, I think, but if so, I don't necessarily remember it. He was well known in the rotifer community. We get together for these gatherings every 3 years or so, and Eric was very well respected within that rotifer family, as we call it. I did, however, while preparing for this lecture, read Martin's excellent and informative celebration of Eric's life in the big newsletter, and I also watched a video of a talk that Eric gave in 2015 to the Queckett Microscopical Society, of which he was a noted member. And I got a sense of getting to know a little bit of his personality just from watching that, full of fascinating anecdotes and memories of the history of the society, as well as his thoughts on rotifers, which were numerous. He made a contribution or a statement that somewhere in his writing about almost every aspect of rotifer biology, and I'll be chipping in some of Eric's contributions during the lecture today. It'll be mostly about rotifers and science, maybe a bit less about taxonomy and records than some of the other talks. That's because there is nobody submitting rotifer records and taxonomic entries. It's such a little studied group. If there's time, maybe a little bit of history too, and some references to Eric's work and contributions. So we'll start with a quiz that seems to be popular in this room this morning. Does anyone know who this is? Sorry?
Van Hook.
Van Hook we have over here. Any advances? Yes? Robert Hooke, yes. Yes, it is in fact Robert Hooke. But you were close to my bait, because the usual answer I get to that is for Leeuwenhoek, who you were thinking of. But this is indeed Robert Hooke. He didn't invent the microscope, but he did do a great service in popularizing it with a book he wrote called Micrographia, where he drew very lovely sketches of things like the legs of invertebrates, sections through cork, and in doing so he was the first to coin the term cells for these things he was seeing at the microscopic scale. We think the instrument itself was invented much earlier in Italy, probably. There is some speculation about Galileo turning a telescope back to front and then looking at microscopic things through it. But the person who's really associated with microscopes and the origin of the instrument is Anthony van Leeuwenhoek, who's a Dutch textiles merchant. And as part of his job in evaluating textiles, he needed to be able to look very closely at the weave and count the threads and so on. So he was very good at building magnifying glasses for that purpose. He had a technique of producing almost perfectly spherical glass globules to look at these details of textiles. But he wasn't only interested in textiles, he also applied his microscope to natural materials. And using those excellent lenses, he was the first to see things that we would today recognize as bacteria, single-celled organisms, and the vast world of microscopic animals, just a scale, an order of magnitude down from most of the work that we've been hearing about this morning. So he would take things like this pond material, and he would pop a little bit onto his needle and look at it through one of these excellent lenses. Here's a later imagining of how he might have looked doing so. Here's the pond scum in the jar next to him. And he discovered a new world. And he drew some of the inhabitants of that world. Really amazing quality considering the time. This is from 1702. These microscopic animals with little feet, and what he called wheels on the heads here, wheel animalcules. This is about 0.5 millimeter long, and he thought these were little worm-like things with wheels or crowns stuck on the heads that seemed to be spinning, and he called them rotifers, which means wheel carriers. You can see how good his drawing was. This is what an actual rotifer looks like with a modern microscope, and it's just captured it perfectly. Here are the little wheels, which in fact are not spinning wheels. They are a field of cilia that beat in a rhythmical pattern to draw in, you can see it maybe here, draw in little particles of food, which is bacteria, debris, the odd yeast cell. The food goes in here, and then it goes down into the jaws here. You can see this, it looks like a heart beating. I think Leeuwenhoek thought it was a heart, but in fact it's the jaws here chomping up those bits of organic debris and turning it into food, which the animal then uses to produce eggs and offspring and so on. And it sticks to the surface using this foot, which is drawn there. What are rotifers, though? So now that we understand the natural kingdom a little bit better with the advent of DNA and modern taxonomy and so on, we can say a bit more about what these rotifers, these wheel bearers are, and how they break down taxonomically. So I'm going to show you a few animal family trees in this talk. You might have seen diagrams like this before. It's using DNA to show the relationship among different sorts of animals. It's a little small, but we needn't be too concerned. This is all the different groups of animals and their relationships to each other. Everything that we've heard about today in terms of animal and invertebrate diversity is all here. Arthropods, all the millipedes and the spiders and so on are all just within there. This is the full diversity of animal life. And rotifers are in a completely different area than insects and other arthropods. They're up here. Where are we? Down here. There we go. Rotifer here. So they're more closely related to things like earthworms, annelids, mollusks. I don't know if there's any snail or slug people in the invertebrate group here, but they're a little closer to the rotifers, taxonomically speaking. We'll just blow up this area of the animal family tree, a bit bigger. And you'll see the sorts of things that are around, again, not close to, but in the rotifer neck of the woods. Mollusks, annelids, ribbon worms, flatworms. Gastrotrix are a funny little group that even fewer people work on. They're sometimes called hairy backs. There's your insects and that up here. And here are the rotifers, finally. And there are several different sorts of rotifers that I'll just deal with very briefly because we want to get to the sort that Eric and I work on. But to be fully taxonomically complete, as I think this audience will appreciate, there are 4 different sorts of rotifers within this group. And they are bdelloidea, the B is silent here, it's just bdelloidea, monogononta, acanthocephala, and Cysonidea. In each of these diagrams, I've given you a typical member of the group and a male and a female, male and female, male and female. Male and female and sex is going to become a bit of a theme of this talk as we go on. So I'm just flagging that now so that you notice the difference. And you might also notice that there's something missing from this top picture. More on that soon. We'll just summarize briefly the natural history of these 4 different groups, and again, we won't spend much time with some of these. The most species are the monogonont rotifers, and these are the ones that I think Eric spent most of his time looking at in ponds and canals and rivers and so on. About 0.1 to 2.5 millimeters long, slightly harder outer body. Most of these are freshwater. There are quite a few marine species as well. They're very important in freshwater ecosystems, provide food for fish, probably for the same food chains that lead to your dragonfly, nymphs, and other things living in the water. Mostly free living, and they're seasonally sexual, so they'll have part of their life cycle being sexual and part of it reproducing asexually. Then we have the acanthocephalans. Now these weren't thought to belong to the rotifers until DNA came along and threw all our old taxonomy into confusion. These are giant, by rotifer standards, giant, 650 millimeters long, parasitic worm-looking things. They infect crustaceans and then they infect fish. And they were their own phylum until relatively recently. And then using DNA, we discovered actually they're just very, very big, highly modified rotifers, these parasitic worms. So now we just call them, well, I call them acanthocephalan rotifers. Cysonids I'll say very little about. They're a tiny group of species that live on the mouthparts of certain marine crustaceans. Again, very slightly modified body plans that we would associate with rotifers. And lastly, the ones I'll be spending most of my time on today, the bdelloid rotifers. Bdelloid means leech-like in Greek. It's because they have a sort of sucker at both ends, so they look a little bit like leeches and they move around in a caterpillar sort of fashion. They are almost all freshwater, or limnoterrestrial, meaning that they live in the film of water on mosses, soil, and occasionally puddles and ponds. Mostly free living, and as we'll hear more about soon, unlike all the other rotifers, they are only known to reproduce by asexual parthenogenetic eggs. And males of this entire group, which is a class, a whole class of animals, is equivalent to, what would that be equivalent to? Is that insect hexapoda? All the insects? Is that right? Class? Is that class? Anyway, it's as diverse and as big taxonomically as all spiders, there you go, all spiders. And no males are known in any of those species. Acanthocephaly, we'll spend very little time with this because it's before lunch and they're a little gruesome, but they are spiny, thorny-headed worms that you find in the guts of fish. Sometimes you might see it if you're a keen angler and you do a lot of gutting. But the fish then drop eggs. So from the guts of fish you get eggs from these worms that then infect things like this shrimp. And then the fish eat shrimp and the worms get back into their guts and the life cycle continues. But yeah, apparently it's a rotifer. They've lost the wheel part, they've got this spiny head instead. But some of the other internal features are quite similar. This is the tree, if you like, of the rotifers. And it explains the reason why I'm talking to you about intestinal worms and funny things that live on the mouth parts of crustaceans. In evolutionary biology, if you want to talk about a group, you have to include everything that's closely related to it. Classically, rotifers were understood as these things, monogonons and bdelloid rotifers, these ones. The problem is that according to the DNA, the tree looks like this. So we've got monogonons, bdelloids, and then we've got parasitic worms things and mouthpart things. And you can't just take these off your tree and stick them together and call them rotifers. That's just not allowed. If you imagine cutting or pruning an actual tree, if you made a cut there and a cut there, these two would be on different branches. You couldn't physically get them together. To make the cut to get them together, you need to cut it here. And if you do that, you have to include the acanthocephalans and these other things, the cysides. So this is why I'm saying this lot are all, if anything's a rotifer, that lot kind of has to be. But we'll move on now and talk about the main groups. So I've drawn attention already to this difference here, the difference between parthenogenesis, that's asexual reproduction, and sex, which we're all familiar with. The main difference between these two groups is that the monogonons have sexual reproduction, at least in part of their life cycle, but the bdelloids apparently never do. Otherwise, they're basically quite similar in most of the other important characteristics. This is a typical life cycle for a monogonon rotifer. They're really morphologically diverse, so once you get the microscope out and start looking at the things, they're really rather different. So you can see this is one species, here's another one, long foot, and some of the details of things like the toes are important taxonomically, the hairs that come out of the head are important. The shape of it's not exactly a shell, but the shape of the outer integument is important, with sometimes spines and things. Some of them will build little sheaths for themselves to hide in and retract into the sheath and then pop their heads out to feed, like this one here. This one's carrying eggs around. Just to go briefly through the life cycle, much of the year they're asexual, so they will lay eggs with two pairs of chromosomes like we have, one from your mum and one from your dad. But they will lay eggs that are exact copies of themselves, just cloning themselves, photocopying themselves for a whole growing cycle. This is just to get the numbers up when they're recolonizing some pond or river after the winter, perhaps. Asex is a very quick and efficient way to make more copies of yourself. And then at some point, when the food starts to run out or the environment starts to deteriorate, maybe it's the end of the season, things are starting to get cold, they will switch to using sex. They'll produce sexual females who will produce males. Males, funnily, only have one copy of all their chromosomes. I don't know if anyone does beekeeping or knows about ants, bees and wasps and how they reproduce, but it's similar to that. They'll produce drones, if you like, at some point, and then they'll go out and mate and produce the next sexual generation. And in the rotifers, these sexual eggs then drop down to the bottom of the pond, and they sit in the mud for years, maybe decades, until conditions are right, and then they hatch back up and start again with the asexual part of the life cycle. So it's a complex mix of sex and asex in the monogonal rotifers. And these were the group that Eric spent most of his time with. So here's a diagram of beautiful draftsmanship and colouring on this. He played his dilatata there that he was doing in 1947. It's just decades of expertise in microscopy. I think he was the youngest person ever to join the Queckett Microscopical Society. And that was before he drew this one. He described new species of rotifers. If you want to describe species new to science, stop with your insects and come and do rotifers. Almost all of them you'll find are new to science. It's very easy to find new species, as Eric did here with this Cachalodella that he found up in the Lake District, I think. Species were named in his honour. This is Norfolka hollidayi. His colleague Herbert Dartnell named it in his honour. This was found down in the Antarctic somewhere. You can still see the slide in the NHM today, if you pop down there and ask. Eric made an important contribution to this, which is the authoritative taxonomic reference for all rotifers. He was given the entire chapter on the family Synchaetidae. I've got this, so you can look at it later if you want to see some of his drawings and his synthesis of other research. Really comprehensive taxonomic contribution there. All on monogonal rotifers, though. So, rather unfortunately, I work on the bdelloids, so slightly different group. So that's the monogonons, and now we'll get onto the real rotifers, in my view, which are these slightly softer-bodied, leech-like rotifers. Microscopic, yet soft-bodied, filter-feeding invertebrates. This is the same animal in three different poses, if you like. This is with its wheels out, wheels contracted, and it's doing this leech-like movement. And this is the animal fully contracted. I think we saw it in that video earlier when I disturbed the slide and it pulled itself back in. Bdelloid rotifers can be found anywhere where there's fresh water on the planet. However temporary that water is, however harsh the environment. So this is like paradise if you're a bdelloid rotifer. This is a stream, some mud, some puddles. Mosses and lichens are covered in water that they love to live in. But you'll even find them where it's stony or dry, where when the water returns, you'll find rotifers there. You can find them on the frozen glaciers of Antarctica. These are the tops of mountains poking out of the top of the ice sheet. And on the, 2 days a year when that ice is liquid or when the film melts, you'll find rotifers, bdelloid rotifers living on the surface there. Deserts, again, it might rain once a year. Plenty of bdelloid rotifers in those pools when it does. And we even found some DNA from rotifers recently in a hazardous chemical mitigation fermenter. So anywhere with there's water, you'll find these rotifers living on the bacteria and other debris that you find in the habitat. The reason is that they can survive extraordinarily stressful environments. They can tolerate desiccation, freezing, radiation, anoxia, starvation, and it's probably because they need to be able to live in these extreme habitats. Like Antarctica particularly, obviously you need to be able to tolerate freezing and being very dry there. In fact, bdelloid rotifers might be the most radiation-tolerant animals of all. Forget cockroaches, even forget tardigrades that are famously always in the press for being the most resilient animals and surviving nuclear war and whatsoever. Here we have some data on radiation, which is measured in grays. We're pathetic. Humans, it only takes 10 grays to knock us out. That's the sort of dose that the immediate survivors of something like the Hiroshima flash would have experienced. Almost instantly fatal. Monogonal rotifers can survive a bit better, so they're still laying eggs. This is fecundity here, even with up to 100, 200 grays of radiation. Bdelloid rotifers, though, they can survive and continue to lay eggs that are viable at up to 1,000 grays. That's what, 100 times more than we could. It's absolutely an astonishing trait, and much more than tardigrades as well. Tardigrades are pretty radiation tolerant, but not as good as these. They just don't get the same press because tardigrades get all of the cute marketing. They get to be called water bears and moss piglets and bear animalcules and other cute things, whereas the rotifers don't get, they don't have little faces and cute bear-like animations. So I'm doing my best to get the profile of rotifers up and putting tardigrades in their place wherever possible. Yes, there are bdelloids. In fact, Eric did make some contributions to bdelloid rotifer biology as well. He went to the Antarctic and, as part of the British Antarctic Survey, produced with his colleague Herbert Dartnell this lovely report in 1985 that mentions 2 solid years of just prospecting in an Antarctic island for rotifers and identifying them and classifying them and so on. Now, it must have been a sort of interesting time to be in the South Atlantic in 1981 to 83, because most people remember the Falklands War was going on at the time. Argentina invaded the Falklands, right? So quite what was going on down there. whether I think the government partly supported the BAS to go and increase activities, to increase the presence down there at the time. I'd love to have talked to him about what he was doing down there and how that experience worked out. If anyone ever did get Antarctic stories from Eric, I'd love to hear that. But from my perspective, the most interesting output was this lovely drawing of this rotifer that I showed you earlier. It's Phylodina gregaria, which is, the most striking thing about it, you'll notice, is this bright orange colouring. And I think more recent work has shown that actually part of that orange colouring is used as sunscreen for the rotifers to absorb the harmful ultraviolet light that they get on them, on the snow, and help them to resist some of that sun radiation. But Eric had a lovely picture of these things in his Antarctic samples. Here's some more pictures of beautiful bdelloid rotifers. Here they are crawling around on a slide, just for scale. This is a hare. It's one of my hares, actually, because I made this slide. These are the animals sort of gliding or crawling around. Here are some parthenogenetic eggs. And here's a contracted animal. They're just very pleasing to look at least to me. Thousands of these animals have been examined over the 300 years since von Leeuwenhoek first reported on them. A lot of it by very keen amateur naturalists like Eric, and in fact another of his important pieces of work was a survey of the last 300 years of rotifer research, who was looking at them and when, and in that time I would imagine thousands or tens of thousands of animals must have been examined. Enough to describe hundreds of species of bdelloid rotifers, multiple genera and families. So again, compare that diversity with your favourite group of arthropods, incredibly diverse. And yet, all those records, all those examinations, not one male has been discovered or detected. I don't think, when I'm talking to academic audiences, they appreciate just how much effort folks like yourselves put into examining closely. We've heard about genitalia, records, how closely these things get examined. And they say, oh, well, they probably just haven't noticed. But if you know amateur naturalists, if you know the effort you put into a species, then it's most unlikely that you would have completely missed a single male in all of these centuries of very close microscopical examination. So we think it's possible that there is no sex and there are no males, and that they reproduce only via these parthenogenetic eggs. One rotifer producing copies of itself and copies of itself, and so on. If so, they would be the largest animal group by far, with no males whatsoever, all entirely female. And that makes them of great interest to us as evolutionary biologists. Funnily enough, Eric had a word, as usual, to say about this phenomenon of all female rotifers. It's in a paper of his describing a report of how to look at rotifers under a microscope. And he rather testily said, now whoever wrote the legends to the photographs in the Armchair Science edition was unaware that all bdelloid rotifers are female, because the legend to this Rotarium macrura picture repeatedly refers to it as a he. Quite indignant. And quite right, he's quite right too. So asexual reproduction, this is the process of just copying yourself or producing eggs that will hatch into basically clones of their mother. It's not impossible, it's quite common in some groups. We think of it as unusual because it never happens in mammals. Mammals are always sexual. As far as we know, we're completely incapable of asexual reproduction. You always have to have male sperm fertilization and so on. Then there are these reports you sometimes read in the news about unusual reptiles or sharks sometimes, that there's a female living alone in her tank, and then one day the keeper comes in and sees there are eggs all over the place, and sometimes they even hatch, and it's virgin birth. So rarely it is possible for some of these vertebrates to go through asexual reproduction. It seems to be a little bit weird in those groups that do it as a last extreme measure when they can't find a male to mate with, but it is possible. But in the invertebrate world, it's a bit more common. So I think we know that aphids, for much of their life cycle, are asexual, copy themselves. Water fleas, dandelions, don't need to pollen to set seed and then produce these clocks with seeds that blow away. All of that is without sex and without fertilization. And then there are a few animals and plants that almost always reproduce without sex, just copying themselves. Bananas, famous example. All bananas you've ever eaten are genetic clones of each other, the Cavendish variety. A few lizards, sea monkeys, and the odd other thing here and there. The puzzle for us is why more things don't reproduce like this, because it's a very efficient way of reproducing. Just copy yourself. And sex is really complicated and seems unnecessarily inefficient. The main problem with sex is the males. You just don't need them. Females would be far better served through evolution to just produce copies of themselves. Why do you need this fertilization step? If you could just copy yourself, then all your offspring would be daughters, and all of them could copy themselves too. Everybody's independently reproductive, and you can grow your population twice as quickly. But instead, you've got sex where you need these males, and half of your population can't put out any eggs. They're not contributing their bit to growing the population. They're just a waste of space from an evolutionary perspective. So that's one big cost of sex. Be more efficient. You just ditched it. Then there's the cost of mixing up genes that work well together. So you've gone through your natural selection and you've got some organisms that work pretty well. They must do if they're ready to reproduce. But right at the minute when they're ready to reproduce and pass on this successful genotype, what do? They get together and mix and shuffle up all of these genes into new combinations that we haven't seen before. And things can go wrong in that process. You have these perfectly well adapted sets of genes, and then you go and mix them up. Who knows what you're going to get? Something far less effective maybe that doesn't work together, hasn't been tested before. And there are other problems. You've got to find a mate in the first place. There are diseases and risks and so on. All in all, it would make much more sense to just be asexual, and yet nearly all animals and plants are having sex. And it's a bit of an evolutionary puzzle. So you do get asexuals. This blue in this tree is asexuals. You might get a species here or there, maybe a whole genus here of asexuals. But they're rare and they're short-lived. You don't get a whole big blue branch of this tree, for instance, that would indicate a long-lived asexual group. Something happens that suggests they're really short-lived and asexuals go extinct quite quickly. And we're not entirely sure why. This has been called the hardest problem in evolutionary biology, why sex even exists in the first place when it's so inefficient. Given these many advantages of asexuality, why are asexual lineages so rare? This problem was first framed by an eminent evolutionary biologist, John Maynard Smith, whom I believe at least one person in this room has met and probably discussed exactly this question. Why sex? Well, without getting into too many of the details, briefly, there are three sorts of flavors of explanation. Over 50 have been put forward, but you can kind of distill them into three main ideas. One is that sometimes bad mutations pop up just by chance. If you have sex, you can fix those bad mutations by bringing together the working parts from the other chromosome. Let me put it this way, maybe it's easier to understand. If you have one broken car over here and the spark plug is gone, and another broken car over here and it's the choke or something else, you can make a single working car by bringing the working parts of the two together, and you can get it to start again. Sex is a bit like that, in that it can fix mistakes that you make in the process by bringing together the working parts. Another idea is that you bring the new benefits, the new inventions together in one genetic background. So anyone who breeds crops might know about something called hybrid vigor, where you bring one variety together with another, and all the best features of those two sometimes come out in the F1s. So if you didn't have sex, you couldn't do that. So bringing together good things, getting rid of bad things. And then the last idea is that whether it's bringing together or separating, sex is always changing and shuffling genes. That's just what it does. Brings them together, you get chromosomes pairing up and then forming new combinations. An important reason you want to form new combinations is to resist diseases that are always changing, and you want to produce a new set of genes that can resist whatever the disease is that's currently affecting your population. So we think it's some combination maybe, or one of these, but we're still not entirely sure, and we'd love to investigate new ways to ask about which of these hypotheses is right. The one I spend most of my time on is the disease one, this last idea. So parasites, pathogens, pests, diseases are everywhere. Anyone who has a garden knows that things will come along and eat your plants, or they'll get sick, they'll get infected. It's just everywhere. And if you are a pest or a disease, the best thing you can do is attack whatever is common in the population now. That's the most abundant source of food. If you focus in on whatever DNA, whatever combination of genes is common, that's where you'll get the most payoff by attacking that. What sex does is it takes whatever's common now and changes it by mixing up the chromosomes, shuffling up the DNA and making something new. And just by virtue of being new, that genotype will be more resistant to whatever the current pathogens and pests are. It's why we do crop breeding. When you get a new disease, you bring in some genes from the wild crops, and that mixing creates new combinations that the parasites and pests can't get to. And in the metaphor for this never-ending race between diseases and hosts is the Red Queen from Lewis Carroll's Alice Through the Looking Glass, because the character and Alice are running in place as fast as they can, but never going anywhere. Much like the diseases and the hosts are changing as much as they can, but they're still locked in this never-ending race. Neither of them can get ahead, because the other one is changing just as quickly. And in this model, asexuals can't break up those genes, because they're just copying themselves. So once they become vulnerable to some pest or disease, if it can get one of them, it can get all the rest too. So the model for this view of why we have sex is called the Red Queen hypothesis, and that's the one I'm interested in looking at. If this is right, diseases help to explain sex, then if you've been asexual for a really long time, you should have problems resisting those diseases. You should be starting to build up a backlog of pests and things that are attacking you. Or maybe you found some unusual other way to keep up with your pests that replaces sex. Here's where our rotifers come back in. So here we seem to have this group that defies this pattern of asexuals being recent and rare. We think we have a whole class of animals where all of them are all female. And they're really diverse. We've got hundreds of species, which suggests they've been evolving this way without sex for millions or tens of millions of years. So they've figured out, somehow, how to live without sex. And this was a bit of a problem for evolutionary theory, because all those theories I just talked about said sex is vital. John Maynard Smith didn't like bdelloid rotifers at all. He found them a bit of a fly in the ointment of his theories. He notoriously said, there's something of an evolutionary scandal. Nothing this should be this asexual for this long. It messes with my maths. And actually, I brought a colleague with me today, a doctoral student from Oxford, Timek Pieszko, who's working on exactly this question. If you want to chat more about the scandal of why rotifers are asexual, he'd be very happy to do that. Just hold up for a second. This is Timek. He's working on exactly this sort of puzzle. For me, I'm interested in this disease angle. So, okay, if they're asexual, how do they deal with diseases? Do they have some other way of keeping up? Well, do they have diseases for a start? Maybe they just don't have diseases. When God was handing out all the diseases in the Garden of Eden, maybe they just got off light. No, not the case. Unsurprisingly, they have all kinds of diseases. Most of them are fungi. So these are rotifers that have been killed by various nasty fungal pathogens. These are the dead rotifers, and these are the fungal spores bursting out with hyphae laden with little particles that will then infect the rest of the rotifers. And these are highly virulent. They can wipe out the populations in just a few days. So there's lots of parasites out there. How do they deal with it? Well, one of the ways we've already kind of touched on. I've said that these rotifers can tolerate complete desiccation for months or even years. When the moss dries out, they desiccate, they form these little contracted balls, and when the water returns, they just pop right back to life. The fungi, on the other hand, while they're in that desiccated state, they can also blow around like particles of dust, a bit like the grains of sand we heard about earlier. They're even smaller than that. They can blow around and land in new habitat patches and start new populations. The fungi, on the other hand, we all know mold likes it wet, so that's why it lives in bathrooms and so on. When they desiccate, the fungus is destroyed and it can't come along with them to these new patches. So the rotifers are able to shed the fungus, move to a new patch and start fresh. And that helps them to survive despite the diseases that they suffer from. You get lots of infections in the wet patches down on the ground level and fewer up in the tops of trees where the moss dries out more regularly. So we think there's a way of escaping from these diseases in space and time by drying up and blowing away effectively. Eric was sort of onto this a little bit. I found a remark he gave at the Royal Society during a microscopy meeting back in 1977. He piped up and said, I think it possible that bdelloid rotifers might lose fecundity if not subjected to alternate desiccation and immersion in temperate regions. And we now think we have a mechanism for this. So not only can they survive desiccation, but if you don't desiccate them, they start to get sick. And that's exactly what we found when we looked into it much later. The desiccation helps get rid of harmful bacteria and fungi that the rotifers otherwise encounter. So that was a prescient remark. So we've got one way of dealing with diseases, escaping from them. But there are a couple of objections to that. Okay, so that helps them avoid the diseases, but what do they do when they actually get infected? Do they have a way of fighting it off? And also, sex is about moving genes around, but this is more about moving animals around, so it's not exactly the same. Is there anything else that they do that might help fill in these two gaps? Yes. So when we started sequencing the DNA of these animals, we had a bit of a surprise. Turns out that when we sequenced the DNA, 90% of the DNA looked like normal animal DNA as we were expecting. But then there was this other 10% that didn't seem to belong at all. So 1 in 10 of their genes didn't look like animal DNA. It looked like DNA from fungi or bacteria or even from plants, completely different from animals as we were expecting. These genes seem to have been stolen from other parts of the tree of life and copied in to the rotifers' genomes. They have been copied from bacteria, most of them. Some of them are from fungi. Some of them are from plants, even. Thousands of these genes in these rotifers are not animal genes at all. They're from some other form of microscopic life. We don't know how they do this. We just know that it's extraordinary. No other animals do this. This is a 10 times higher rate of gaining genes horizontally than any other animals. Most animals just have normal animal genes. So something very strange is going on in these things. What sorts of genes are they stealing? And might that have any connection to the challenges that they face? We recently found that one of the most important groups of genes that they steal are these giant enzymes. This is now getting a little technical. Giant enzymes that are called biosynthetic gene clusters. You can think of them as like molecular chemical factories. They are genes that churn out chemicals, and the kinds of chemicals that they churn out, you don't need to understand the organic chemistry, but they're antimicrobial chemicals. They are things like antibiotics, antifungal compounds, chemicals that kill other microscopic life. These genes that do this are extremely common in bacteria and fungi, but really rare in animals. Animals don't do this. However, it's the same sort of genes that we use to get things like penicillin and other antibiotic and antifungal drugs for us to use. So the rotifers, it looks like, have been stealing from the bacteria and the fungi in the soil the genes to make antibiotics and antifungal chemicals, and then using those against their own enemies. And they can use these to resist the diseases. So here are two rotifers that we attack with the same fungus, by just dumping a load of fungal spores onto the rotifer, same number of spores onto a different rotifer, we found big differences in which rotifer was resistant and which one wasn't. This rotifer, Aninetta vaga, was very susceptible. 75% of these animals were dead after 2 days. Whereas this rotifer, Aninetta ricci, most of the animals survived, only 15% were dead. It resisted them. And when we looked at what it was doing to resist the fungus, this resistant animal, to simplify quite a bit, we found, and again I'll skip through this because we're in the interest of time, but we found that the biggest difference in the genes of these two animals was this set of resistant rotifers were expressing antibiotic genes that they'd stolen from bacteria. These chemical cellular factories, and they were churning out antifungal chemicals using these stolen genes. So we think they're gaining protection from parasites using stolen genes. Microbes make these genes to kill each other. The rotifers take up the DNA from the microbes, copy them into their own genome, and then they can make these antimicrobial chemicals and use them to protect themselves from fungi. And if you want to read more about that, we had a little popular article in The Conversation last year about our work on rotifers and antimicrobial chemicals. So now we have two exciting kind of ideas as to how they can deal with diseases. They can escape from them in space and time by drying up and blowing away, and they can steal genes from other microscopic life to fight their own fungal diseases. So just to summarize, we have these funny little creatures that don't look like much, but are actually the largest group of animals that have managed without males or sex. They also have these two tricks that help them manage diseases. They can escape from fungal diseases by drying up and blowing away, moving themselves around instead of moving genes around. And they can steal recipes for medicine, effectively, that they turn on when the fungus attacks. So they're also sort of moving genes around. They therefore give a kind of clue as to why sex might exist. Is it just a coincidence that this one thing that doesn't have sex also has these funky tricks to deal with diseases? Maybe not. Maybe they don't have sex because they have these other tricks to deal with diseases. Or, if you're not having sex for a long time, you do need some other way of dealing with diseases, and they've found a couple. We're now interested in sort of using the bdelloid rotifers for our own benefit. So if they've been around in the environment scavenging antibiotic genes from different sorts of bacteria and fungi in the soil, what have they found that we might not have found yet? We're doing the same thing to find the new drugs to fight antibiotic resistance. What we're now looking to do is cut out the middleman, if you like, and just go to the rotifers and ask, what have you found in the millions of years of feeding in the soil and copying genes? Have you found anything useful? It might be especially handy because they're animals, and so whatever they've picked up must be sort of safe to use in animal cells. One of the big problems with discovering new antimicrobial drugs is they're often toxic to us, to animals. That's why we have clinical trials and so on. So if we can figure out what they've taken and make that, maybe it'll be a leg up on the next generation of antimicrobial and antifungal drugs. And then you can read more about any of these stories using any of these QR links, if you like. Now I was going to tell you a different story about taxonomy, but I don't think we have time. I will be around later to talk more about the taxonomy of the rotifers and the fun story, and I have a poster on it too that I might stick up maybe next to Eric later this afternoon. I do, however, want to skip forward to my final acknowledgements. It's a fun story. So yes, I wanted to just bring to Eric's community here in Buckinghamshire the memorial that was written by the rotifer family. What the nice things they had to say about him. So in their report from their recent meeting, they recognised Eric and they said, well, we said, we want to show deep thanks and respect to these colleagues who produced great advances in our understanding of rotifer biology and ecology, and unfortunately are no longer among us. Where we stand on the shoulder of these giants, and Eric was one of those in the rotifer community, following in a tradition of microscopists and natural historians going right back to Leeuwenhoek and so and to I recommend a few of Eric's papers about rotifers that are just very engagingly written and very much worth picking up and having a look at. Yes. All right, I think that will.
Thank you for
Are there any pressing questions? Or are you desperate to break for lunch? Oh,
Have we been in space?
Yes. Yes, like the tardigrades, they've sent some up. I don't know quite why. The European Space Agency loves to just send things into space. It's partly just to justify having a space program in the first place. But yes, they've been to space. No, we didn't learn anything particularly exciting or new from the fact that they did survive the space environment.
oh yes, there's a question over here.
Are they still taking in new fungal genes?
Yes.
Like way back ancestors?
Lots of it is way back, but we did find one that seems to have come in very recently because it looks an awful lot like a current modern bacterial gene in various ways. So they're still doing it, yes.
Can you put a slide up with the references to the articles?
Yes, of course. Sorry, the articles.
These
Oh, mine, yes, not
the
Eric's.
articles.
Yes, we'll have to go back a few
Sorry, I'm
bits
sorry.
of that. That's fine.
Yeah.
So the QR codes should give you links. There we go. Links to those 3 stories. If you take a picture of it, you can later blow them up and work whatever magic with the codes.
One further question. David.
Have any been resurrected from ice cores, permafrost, that kind of thing?
So there was an article that got an awful lot of attention about 3 years ago from a group of Russian rotiferologists who made the surprising claim that they'd resurrected a rotifer from 23,000-year-old permafrost. That's previously the record of reviving frozen rotifers was 12 years. To go from 12 to 23,000 raises some eyebrows. We're skeptical of that report, but you will find it cited in lots of prominent places. It was a peer-reviewed paper. I didn't review it. Well, yes, I'm skeptical of claims of quite that amount of longevity, but yes, they probably do exist in more recent permafrost cores.
Right, we are going to break now, but thank you so much for coming over and introducing a sort of different level of presentation to us. And it's fantastic to have this, especially when a lot of us knew Eric, who would sit in the corner of our meetings and often fall asleep. And then suddenly he'd wake up and offer a really erudite comment on something that was going on. So it's wonderful for us to be able to remember him. So thanks again.
Thank you. Thank you for asking.